In today's topic, I will deal with circuits, connections, and functions of cerebellum. Here I have made a schematic diagram of the circuits of the cerebellum, which I would be discussing during my presentation. Look here, this is the cerebral cortex, and this is cerebellum. The cerebellum is 1/10 of its mass, or however, if you are looking at the number of neurons present here and number of neurons present here, is definitely much different. It is 4 times. It is 16 billion neurons here, entire cerebral cortex, and it is 69 billion neurons. That is about 4 times this. Okay, this one I'm quoting it from this reference. So this is possibly, or this is possible only because of the folia and the fissures the cerebellum has. This is the part of the hindbrain, and it has a folia and fissures, and it has a enormous, or 4 times greater than this, greater neurons than the cerebral cortex, and this hindbrain component is attached to the medulla and pons with the peduncles, the superior, middle, and inferior peduncles. Now, these are just to revise what I have told in my previous class. This is a cerebellum. This is the leaf-like structure, a functional component of the cerebellum. Okay, and this functional component of the cerebellum composed of the lateral part of cerebellum. This is the lateral cerebellum, and the intermediate part of cerebellum. This is intermediate part of cerebellum on either side, and this is central cerebellum. The intermediate and the central makes the spinocerebellum, and the central part is called vermis, and this is connected with the posture regulating mechanisms, or the axial muscles. Then we have in the bottom the flocculonodular lobe. This is the flocculonodular lobe, and this is connected with the vestibular nuclei, and this is a vestibular part of the cerebellum. The cerebellum thus is divided into cerebrocerebellum, the spinocerebellum, vestibulocerebellum. And the cerebrocerebellum is connected to the cerebrum, that is through the cerebropontocerebellar connections, that is the input. So then this vestibular cerebellum is connected with the vestibular nuclei and other inputs coming from the reticular system. Then we have the intermediate cerebellum. This is the intermediate cerebellum is connected with the spinal cord, that is a dorsal spinocerebellar tract, a ventral spinocerebellar tract, a cuneo-cerebellar tract. So these are all coming here, and this is the central part. It is connected with the tectocerebellar, reticulocerebellar. Okay, each of these cerebellar parts have a deep cerebellar nuclei. We have practically 3 deep cerebellar nuclei, and these are now called as a vestigial nucleus, interpositus, dentate. A dentate nucleus is present in the lateral part of the cerebellum, or the cerebral part of the cerebellum. The interpositus is in the intermediate part of the cerebellum. It has 2 nuclei, nucleus emboliformis and nucleus globosus. And this central part is a vestigial nucleus. And the flocculonodular lobe shares the vestibular nuclei in the medulla. So thus there would be, these are the nuclei. If you are looking out the output pattern, this vestigial nucleus provides output to the medial descending system. This medial descending system is governing the axial muscles. So I have made it here axial muscles. That is for the axial muscle contraction. The intermediate part of the cerebellum through the interpositus nucleus will be talking to the lateral descending system, and this lateral descending system is to the distal muscles, that is for motor execution. So this is for execution. And now the dentate part, the dentate nucleus, this is the dentate nucleus, is connected to the cerebral cortex, the motor cortex area 4 and 6. And this is for motor planning and motor sequencing, all the cognitive components of the cerebellar motor functions. Then the vestibular cerebellum or the flocculonodular lobe is connected with the vestibular nuclei, and also it is connected with the collicular nuclei or the tectal nuclei for the balance and the eye movements. This is in brief about the parts, the nucleus, the functions or the output of the cerebellum. I have in this I have covered 4 objectives: 1, connections, the parts, connections, the nuclei, and the output of the cerebellum. Then move on further. The cerebellum has a functional unit, and this is the functional unit of cerebellum. What is this functional unit of cerebellum? Now, the cerebellar cortex is divided into 3 layers: the outer molecular layer, the Purkinje cell layer, the granular cell layer, and the white fibers, and then deep cerebellar nuclei. That is the cortical structure or the histological structure. Now, what is there in this box is the unit of the functional unit of the cerebellar cortex. And we have such functional units, nearly 30 million of such units in the cerebellum. You can just imagine. And so this red one here, the red part is excitatory, black one, this is a inhibitory, and the blue one is a output neuron. So now we have the same functional unit I have brought in here. I have just brought. So it has 3 components: input components, the output components, and the modulatory components. What are the input components? The input components are the Mossy fibers. This is the Mossy fiber that is coming from the several areas of the brain, brainstem, the spinal cord. So this is a Mossy fiber. And then granular cell, because this provides input to the granular cell. And then it will make the parallel fibers. These are the parallel fibers. And then it will, because of these parallel fibers, interact with the Purkinje cell dendrites. This is the Purkinje cells become the part of the input component, that is especially dendrites or dendritic tree. So then the another input device is a climbing fiber on the Purkinje cell. So the Mossy fibers reach the Purkinje cell indirectly through the parallel fibers. The climbing fibers directly encroach the Purkinje cell or embrace, intertwine the Purkinje cell. The output components come from the Purkinje cell. This is the output. Then we have the deep cerebellar nucleus. This is the deep cerebellar nucleus that gives the output. Then we have a third is the modulatory components. The modulatory components here in this picture are not shown. I will just mention the Golgi cell, which checks the granular cell activity, the stellate and the basket cells, which checks the Purkinje cell activity. Maybe here you see those. This is a Golgi cell, and these are the basket cells and Purkinje cells. Now, so this is again this picture of the basic circuitry of the cerebellum. What I started from here in the functional unit, now because it is missing certain cell types, that is why I have just added up these cell types. Also basket cell and stellate cells, you can just see that outer molecular layer, Purkinje cell layer, granular layer, white matter, and brainstem or spinal cord. Now come back here in the you have this mossy fiber. This is a mossy fiber, and here in this we have three layers: outer molecular layer, Purkinje cell layer, granular layer, white matter layer, and the deep cerebellar nuclei. That is what we have marked here in this particular thing. Then we move on. So the cellular, what are the cellular elements of these circuits? So in this circuit, so we have 6 neuronal cells. Of course, there may be n number of or many more glial cells, which we are not. The only neuronal cells, cell types are included. So they are Purkinje cell. This is the Purkinje cell. You can just see that this is the cell body there, and this is the dendritic tree, and this is the axon reaching to the deep cerebellar nucleus. So this is one Purkinje cell. Here in the bottom, I have mentioned one typical Purkinje. It's a real picture of silver staining, and you can just see that how intricate these dendritic arborizations are. You can just see about that. This is the cell body of the Purkinje cell, and these are the dendritic arborizations, and this is the axon, which is reaching to the deep cerebellar nucleus. This is the Purkinje cell layer. These are granular cells. They are very abundant cells of the cerebellar cortex. So that means for every one Purkinje cells, there may be many granular cells. And the specialty of these granular cells, they have a few basal dendritic things, and they have a apical projecting axons which bifurcate as T. You can just see that you have these these are parallel fibers. These axons become parallel fibers. You can just see that these parallel fibers, maybe we have shown only 4 of them. So that means for each there would be many more granule cells. That means entire this outer molecular layer is having these parallel fibers. Now, if you are looking at this particular Purkinje cell there, and if you cut these with the parallel fibers, you can just imagine how many cuts it makes, and at each cut, at each intersection with this Purkinje cell, there would be a synapse, and that is the information the granular cell tries to provide to the Purkinje cell. Now, then after the granular cell, there are basket cells. This is a basket cells. It is in the almost in the little deeper aspect of the molecular layer, and this one will have it has a contacts with these parallel fibers, or synaptic contacts with these parallel fibers, and its output, the axons, reach to the soma of the Purkinje cell. Then we have the stellate cell. Now you have the stellate cell is the most superficial or the external part, external cell. And these stellate cells are also, again, these dendrites are cut by this, and then axons reach the dendrites of these Purkinje cell. So that means they are only dendrites here. It is with the soma. That is a stellate cell. The Golgi cell, this is a Golgi cell, and this Golgi cell, what happens? It checks the activity of the granular cell. This is one Golgi cell, and this thing, and this is the activity of the Golgi, the granular cell. Then the deep cerebellar nuclei. This is the deep cerebellar nuclei. It receives inputs from the mossy fibers and climbing fibers, and the input from the cerebellar Purkinje cell, and then the output goes here. Output is either it is excitatory, either it is more excitation or less excitation. This is about the cell types. Then fibers. So it has three major fibers in the that's the axonal fibers here. One is the Mossy fiber. Mossy fiber. This is the Mossy fiber coming from the various locations of the brainstem, spinal cord, vestibular system, colliculus, red nucleus, and the pontine, that is the corticopontine fibers. And all these things, they first they give information to the deep cerebellar nucleus, then to the granular cell, and these granular cell would make the activation of these dendritic tree. What I have missed here, the Mossy fiber is also making contact with the Golgi cell. So it will also have a synaptic contact with the Golgi cell. A collateral reaches there that is not shown. Okay, so now this Golgi cell in turn projects onto the dendrites of the granular cell that checks the activity of the granular cell. One maybe because of the direct activation here, and secondly through the indirect activation. This is about the Mossy fiber. The outcome of the Mossy fiber is the stimulation of the number of cells or number of dendrites. One is Purkinje dendrites, the many Purkinje dendrites. Then it will stimulate the number of stellate cell and basket cells, and it will stimulate Golgi cell. And the Purkinje cell, when they are stimulated, the output is reaching to the deep cerebellar nucleus. And the activation of these stellate cell and basket cells, what they do, their output, because it will excite them, and the output of these things is inhibitory to the Purkinje cell activity, and that I mentioned as a feed forward inhibition. So that means first, because it is activating the Purkinje cell, and this Purkinje cell is giving here, even before the arrival of the Purkinje cell activity, they are trying to inhibit it. It is for focusing or sharpening of the activity of the Purkinje cell. So that is about Mossy fiber. A climbing fiber originates from the inferior olivary nucleus, olivocerebellar tract, and this one is directly in one. It gives contact. It makes first. It excites the deep cerebellar nucleus in the initial stage, followed by it excites the entire the Purkinje cell, because it has a number of contacts, maybe 300 to 500 synaptic contacts in this tree. In this tree, this is the tree there, and you can just see that this blue one is going all through. So this is about the climbing fiber. It stimulates strongly the Purkinje cell, and it brings about a abrupt change. So by the way, the Mossy fiber activation acts as a slow break, and the climbing fiber is a hard break. So it is a sudden, abrupt break in the activity, because it activates this Purkinje cell. The Purkinje cell activation inhibits the deep cerebellar nucleus, and deep cerebellar nucleus for some time it will be less. It will decrease the frequency of activation. So I described about the parallel fibers, or the axons, or the T axons of the granular cell, which intersect all these things and which may activate the Purkinje cells, the basket cells, the stellate cells, and the Golgi cells. And these synaptic contacts are glutamatergic, and they will stimulate. This is about the fibers of the things. Now comes the another important aspect of the whatever I have described. I am going to summarize here. Cerebral functional units contain 30 million such units. This is the unit I have already described. Mossy fibers input comes from various parts of the body, various parts of the brain. So these are the sources of the mossy fiber. Corticopontine from the cerebral cortex, the premotor area, to the pons, the red nucleus, the rubrocerebellar, vestibulocerebellar, colliculocerebellar, reticulocerebellar, spinocerebellar, the dorsal and ventral cuneo-cerebellar activity. These are the mossy fiber inputs. These come from the various parts that excite initially, that excite the deep cerebellar nucleus. This is deep cerebellar nucleus. This excites this. Then granule cell projects the parallel fibers. So now it will then come here and it will excite these granule cells. So this is where it is making the granule cell, and the parallel that will they will form the parallel fibers. You just see that the parallel fibers and in T axons. Several million parallel fibers have 80K or 200K connections with each PC. Can you imagine that? Several million parallel fibers are there, and they have nearly 80,000 to 200,000 connections. So you can. That is why I'm just trying to post this particular Purkinje image. So Purkinje cell image, and it is a true image of a Purkinje cell from a silver staining. So now you can just see that it will cut one parallel fiber cuts many, and if you have several of them, so that would cut many things, and that would be maybe around 100 to 200,000 synaptic contacts with each Purkinje cell. So that is enormous. However, the activity of the mossy fiber is not strong. It is only small, tiny activity. And these tiny activities are summated here in the initial part of the neuron, that is the initial segment. So here, and even when that is happening, we have other cells functioning and trying to inhibit that. That is the feed forward inhibition. Purkinje cell to granule cell ratio is 1:500 or 1:1000. So that means for each Purkinje cell, there would be 500 to 1000 granule cells are there in the cerebellum. So that you can just imagine how complex the cerebellar structure and the architecture is. The mossy fibers produce simple spikes. I said they make only single action potentials. Here is an example of the mossy fiber activity. You can just see that the single action potentials are there, and this is the recording of electrode. Recording electrode is placed over the Purkinje cell. This is the Purkinje cell activity. When they have placed the microelectrode in the Purkinje cell and recorded, and these are having a spontaneous activity, that is simple spikes are coming, maybe around 60 to 100 hertz or something like that, and that is coming here. That is because of these the simple activity, and this simple activity is summated here. This would be about 60 to 100 hertz frequency, simple spikes. There is one more activity, the climbing fiber activity. So that would be known as a complex spike. I will describe about a complex spike in the subsequent one. This is one of those references. You can just see that here I wanted to mention that this is the input. This is the mossy input reaching the deep cerebellar nucleus, and then activity is coming. So that is how it's coming up. So then it will stimulate the granular cells. Granular cell activates the Purkinje cell dendrites, and then it comes back. So this is the circuit. This is the circuit. So that means this is one direct here activation followed by the activation through the granule cells. This is one part. This is simple part. This is simple. Then another is there, and this one is not efficient. So then the inferior olivar nucleus sends impulses that would activate the Purkinje cell, and that would inhibit or regulate the activity. Now, we move on the cerebellar functional units regarding the climbing fibers. Because I was trying to talk about the climbing fibers, let us now consider the climbing fiber. The climbing fibers originate from the inferior olivary nucleus. These are olivocerebellar tract. Surprise, each of these supply several deep cerebellar nuclei. These are deep cerebellar, several of them, it will supply several units of the deep cerebellar nuclei. And nearly 5 to 10 Purkinje cells. Each of these have a collaterals. It will have a 5 to 10 Purkinje cells that would handle. So with each Purkinje cells, it makes more than 300 synaptic contacts. And activation of the climbing fiber, this activation or a stimulation of this thing produces a complex spike. And this complex spike is having a long duration, more than 1 second, or a strong inhibition of the deep cerebellar nuclei. Now, if you are looking at this, the electrical activity, if you are looking at, so this is the inferior olivary nucleus, the activity, it is coming here, and it stimulates the deep cerebellar nucleus. The activity reaches here, and at the same time, because this would go to the output, the part of it is reaching the collateral will reach the inferior olivary nucleus. Inferior olivary nucleus wants to know everything. So now from here it will stimulate here. When it is stimulated, it produces the activation. You just see here this red pink portion. The pink portion is the complex spike. You have this activity prolonged, and after the activity there is a depression. There is no activity. Same thing happening here. This is the activity of the deep cerebellar nucleus. When the electrode is placed in the deep cerebellar nucleus, this is the activity, and it corresponds with this pink area. There is the abrupt stoppage of the impulse activity. Now, if you are looking at the inferior olive at this point of time, when the start of the complex spike, there is a discharge. So that means at this point, the inferior olive gives the activity. So when this gives the activity, that produces a abrupt, large spike, complex spike, and a prolonged potential up to 1 second, and it strongly inhibits the deep cerebellar nucleus. This is a complex spike. Now, again, revisiting the basic circuitry, because this is the basic circuitry here, with already I have explained. The Purkinje, I start with the Purkinje cell. The Purkinje cell have arborization. This axonal output with a deep cerebellar nucleus, and the deep cerebellar nucleus gives output, and the Purkinje cell receives input from the granule cells through the parallel fibers, and the granule cells receives input from the Mossy fibers. And these Mossy fibers, they receive the granule cell activity, and when they are activated, they will go and activate these Purkinje cell dendrites, and they will try to sum up, and they will try to inhibit the deep cerebellar nucleus. Now, simultaneously, they will also activate a stellate cell and the basket cells, and these stellate cells and basket cells produce a feed-forward inhibition. They also thought to be the lateral inhibition. Now, the same thing I have made here. This one is the Purkinje cell, and this is the Purkinje cell I have made. This is the Purkinje cell. A beak indicates the dendritic tree, and this is the axon reaching to the deep cerebellar nucleus. And this Purkinje cell receives input, Mossy input from the granule cells. These granule cells have these parallel fibers. These are parallel fibers. Same thing, I'm just trying to bring the corollary. So now these Mossy inputs, then we have the climbing fibers reaching to the, say this is climbing fiber reaching to the Purkinje cell. Here the climbing fiber reaching to the climb. Then we have the stellate cell. This is a stellate cell here. This is a stellate cell. And we have the basket cell. This is the basket cell here, and this is the basket cell. Then we have the Golgi. These are granular cells, and we have the Golgi cell. This is the Golgi cell, and this is the Golgi cell. These are the various circuitry of the cerebellum. Let us consider them, the chronology of events in the cerebellum during an action. Suppose if a cerebellum receives an input, and what is happening and how it will happen, what are the various cells activated. Let us start with our action begins with the input from the Mossy fiber. The Mossy fiber input comes from the various parts of the brain, that is the brain stem, the spinal cord, the cerebrum, the red nucleus, so on. The Mossy inputs arrive in various parts. So this is the Mossy input, the number one. I've just numbered them. Number two, excite a deep cerebellar nucleus to produce a response. This would produce, or this will stimulate the deep cerebellar nucleus, and that would produce the response. That means that it enhances the activity. And when it produces an activity, so then at the same time it will reach to the granule cell. These are granule cells. And these granule cells, when they are activated, they would reach to the Purkinje cell dendritic arborization. This is dendritic arborization, and they would excite the dendrites of the Purkinje cell. And in turn, they will activate the Purkinje cell. When the Purkinje cell is activated, that would have a output to the DCM. Now, so that means they then excite granule cell, which in turn activate Purkinje cell via parallel fibers. This is Purkinje cell via parallel fibers. Number 4, you can just see number 4 here. Now, so 4A and B, these are modulations. 4A is active because of these things. This makes a contact with the satellite cell SC, and that would be inhibitory to this feed forward. And the basket cell, this would be the basket cell there, and that would be inhibiting the Purkinje cell. So that means this is a feed forward inhibition it provides. Then Purkinje cell output inhibits it suppresses the deep cerebellar nuclear activity. So it releases GABA. This releases GABA here. It is glutamate here, and it is glutamate here. The granule cell secretes glutamate, and the Mossy fibers, they are releasing glutamate excitatory transmitters. The remaining are GABAergic. All are GABAergic. So this output is GABAergic. The output of the basket cell is GABAergic. Output of the Golgi cell is GABAergic, inhibitory. Output of the Purkinje cell is GABAergic. So thus what happens is dampens the action. Okay, let me tell. So now the Mossy fibers come and activate. There is a contraction of the muscle. And once it is activated, this contraction has to be steadied. So it should go slow, and it should not overshoot. It should not overshoot like this. What happens is simultaneously it will activate the granule cells immediately after that, and all of that activity is summed up, and it is coming here so that the overshoot is prevented. If the overshoot is prevented, the dampening or it suppresses the overshoot activity. Usually our actions are, if cerebellum is not there, so these are pendular. So that means they will be oscillating after. So that means they keep on moving like this. So now this oscillates the overshoot or undershoot or overshoot mechanism, so that it will be within the fine tuning or fine activity. This is the chronology of activity events in the cerebellum during one part. So we have taken only the Mossy fiber. Mossy fiber output, when it is going, that will also provide an input to the inferior olivary nucleus. Now the inferior olivary nucleus is monitoring the activity, because the inferior olivary nucleus, the chart from the cerebral cortex that this action has to be done this much, this is the sequence. Now the Mossy output is not matching. If it is not matching, what happens? The inferior olivary provides the excitation. The output of the input inferior olivary nucleus is coming through the climbing fiber. This is the climbing fiber. And this climbing fiber, first it elicits the deep cerebellar nucleus. So inferior olivary nucleus sends inputs to the climbing fiber and excite the deep cerebellar nucleus to produce a response. Then excite Purkinje cell strongly. It will excite the Purkinje cell strongly. And then if it is excited strongly, what happens? It abruptly inhibits the deep cerebellar nucleus. So thus it produces the inhibition of the deep cerebellar nucleus and produces a abrupt interruption of the action, because it halts the action. And that is how this correction, the chronology of events in the cerebellum for correction. So what I mean to say that, suppose I have discovered I am trying to lift a weight or an empty box, and when I reach there with a large amount of force, so this force copy, whatever the empty box force copy, has been given to the inferior olivary nucleus, also to the spinal cerebellum. When it comes, the Mossy fiber unit comes and does the work, and then what happens, it will also get inputs from the muscle. They will be correction. This correction is done by the climbing fiber or inferior olivary nucleus. Okay, that is a correcting action or corrections. Now we move on. So what is that voluntary activity? Just let us have the total view of the voluntary activity. The impulse is from the premotor cortex in reference to the idea. So whatever the idea, pass or reach the lateral cerebellum. Usually they also reach to the basal ganglia, which we are not, right now we are not taking care of that, because we discussed about that. So now this premotor area that will reach to the cerebellum, especially the lateral part of the cerebellum. The cerebrum is connected to the lateral part of the cerebellum, and these inputs are Mossy inputs. The lateral cerebellum provides the plan of action, because now in the lateral cerebellum already, that means as a process of learning, when we are trying to do the motor activity, it will learn what are the muscles to be involved, what are the distal muscles to be involved, what are the axial muscles to be involved for each action. They are all sequenced. These particular things, they sequence them, that is one after another, and they coordinate them. That means it will start doing that flexion or this thing. Then the position required for that particular reference also is provided, and even sometimes the anticipatory components are also provided. Here I tell you, these anticipatory components, we all nowadays use the mobile. And in this mobile, what happens, we have a very small, tiny mobiles, and the particular area, we have to hit on icons or letters in a small space and small thing with the finger. And when you are trying to do that, you have to hit it with a particular force and a particular timing. You cannot keep on pressing there, and you just hit and come back. And these type of movements are known as a ballistic movements. That means typing and coming. That is a touching and going, touching and going. So that is a typographical, or just like a typing. These are called ballistic motions. And in these ballistic motions, there are anticipatory components, because the corrective measures are not possible here in the ballistic motions. And in this, the anticipatory correction is already built in, that we develop, and that is developed in the cerebellum, lateral part of the cerebellum. Now the dentato-ponto-thalamo-cortical projections, that means this is the projection there, from the lateral cerebellum, they will reach to the motor cortex, and from the motor cortex, it will come to the corticospinal tract. That is the whatever the distal muscles or the proximal muscles, whatever. So then the input also, this also receives input from the basal ganglia. Now the basal ganglia and lateral cerebellar inputs, they are sensed here. They try to, basal ganglia tells something very interesting, and this lateral cerebellum tells something very interesting, and it will sense and take the gist of the output given by the basal ganglia and lateral cerebellum, and which is useful or which is essential for our particular activity, that is taken care of. The basal ganglia and lateral cerebellar inputs are sensed, and they give the exact plan, because they give here whatever is come here in the corticospinal tract, it is the exact plan. It is all titrated. It's all titrated and put together. So that is about the overall generalized plan of the voluntary activity. Now, move on. The cerebellum as a comparator. Now, let us study. This is another complex situation. So then what happens? The motor cortex, this is the motor cortex here. The motor cortex sends the intended plan to motor neurons of the spinal cord through the corticospinal tract. So this is coming to the corticospinal tract, to the spinal cord and to the muscles. Spinal cord and to the muscles. So now this is the intended actual plan, that is what is being thought by the motor cortex. This is how it should be done, intended plan. Now the carbon copy of the intended plan is also sent to the intermediate lobe of the cerebellum. Intermediate lobe of the cerebellum, this carbon copy. So you just see that this is a carbon copy. It is coming here. Well, up till now, you are clear. One is the corticospinal tract descends down with a definite plan of activity given by the motor cortex. You all agree with that. Now the same plan, or a carbon copy of the same plan, what I say intended plan, so is also given to the intermediate part of the, or intermediate zone of the cerebellum, that is the spinocerebellum. That's fine, exact. Now plan received by in the spinal cord, it receives the plan. That motor neurons, the alpha motor neurons receive the plan. Okay. Now the collaterals reach to the ventral spinocerebellar tract. Collaterals reach to the ventral spinocerebellar tract, and these collaterals, because now what they do is, yes, we have received, this is the plan. This is the content of that plan, or whatever. So now plan received in the spinal cord is sent to cerebellum through the ventral spinocerebellar tract. This is the ventral spinocerebellar tract. What happens? This is because whatever is received here, it is just sent as collaterals. Okay, this is what is received, and through the ventral spinocerebellar tract, again it is given to the intermediate zone. Now two things are happening. Intended plan is going down. Carbon copy is given here. Then whatever the plan received in the spinal cord is provided through this blue line through the ventral spinocerebellar tract. Now that is up to that thing, it is fine. Now there is a fourth component. Whatever plan received in the motor neurons is different than the plan performed by the muscles, because the muscles have to perform, and the muscles perform the exact action is performed by the muscles. And this exact action is assessed by the proprioceptive components, the proprioceptive senses. Now the proprioceptive inputs coming from this area of the muscles, they will ascend up in the dorsal spinocerebellar tract, and this is exactly what muscles have performed, this proprioceptive action. So now we have a intended plan, it is the executed plan. So this is execution. So the exact action performed by the muscle is provided by the proprioceptive inputs from area of the cerebellum, via dorsospinocerebellar tract and posterior column tracts. This is a dorsal spinocerebellar tract. This will reach here. Okay. In the cerebellum, the intended plan and executed plan, this will provide the executed plan, and this one is giving the intended plan and a carbon copy of the intended plan, and the intended whatever the information submitted for the intended plan, and all these three inputs are processed and compared in the cerebellum. Okay, if they are falling short, if they are not matching, here, so then there will be a correction, as the correction is brought about by the inferior olivary nucleus through the olivary tract. Okay, another interesting thing happens from the spinal cord. There is a spino-olivary tract. The spino-olivary tract. So this will reach to the olivary nucleus, and this inferior olivary nucleus that would be reaching the intermediate zone of the cerebellum. Look here, just for our clarity, I have included the olivary nucleus on the same side. In case of the olivary nucleus, it is on the contralateral side. It's on the contralateral side. So for our understanding purpose, I have placed it here, but it is from the contralateral olivary nucleus, opposite olivary nucleus, that would project on. Now, the green line is olivary nucleus, and this olivary nucleus receives input from all these areas: the cortex, that is the motor cortex, from the collicles, that is the tectum, that is about the eyeball movement and eye movement, from the red nucleus, rubro-olivary, thalamo-olivary, thalamus, the ponto-olivary, reticulo-olivary, vestibulo-olivary. All these inputs, they will reach the olivary tract, and that would give to the intermediate lobe of the cerebellum. This is the plan, and this is the one which corrects, because whatever this would match here, and if it is not matching, it would correct. If it is happening smooth, it will keep quiet. It will not do any correction. So now this, whether it is to be done or not to be done, it is the process of the learning. Learning in the sense, when we try to do an action wherein the inferior olivary nucleus checks our activity very often, whenever I want to do something, so that is in the learning phase. Later on phase, everything becomes the motor cortex also knows how much to give, and this whatever the action, all things should be matched. Then olivary nucleus does not it will not be as much activity. That is what is called a plasticity, or learning. That is what the cognitive component of the motor skill development, because now I am processed, everything is perfect, I am doing the action as expected. So that is what this thing indicate. That means inferior olivary nucleus provides the corrective measures. So now in the cerebellum, the intended plan and executed plan are compared, and if not matching, the correction is brought by the by reinforcing the red nucleus and motor cortical. Now what happens? The inferior olivary nucleus sends information, and this sending of the information through the climbing fibers. So that means it inhibits. So once it inhibits, the number of the activity pattern here, red nucleus and other things, or inhibition or no inhibition, that would be enhanced here, red nucleus and the cerebral cortex. Then that is how they bring the corrective measures. That is how it prevents the overshoot. That is how prevents all those pendular activity. Now, just to summarize what I am trying to mention you all. So first part is the planning of the action. The planning of the action is done by the premotor cortex in association with the lateral cerebellum. Here, the sequencing, timing of the group of muscles, planning is done here in the lateral cerebellum. Now, this information is provided to the motor cortex, and from the motor cortex, it comes back to the premotor cortex. And also it gives the information to the brainstem. Here I'm mentioning the brainstem because the part of it is going to the olivary nucleus, part of it is going to the reticular system and the vestibular system for the posture regulating mechanisms. So that is about the planning. So because this copy of the information is sent, the copy of the information is sent. Come on, I have sent this information. Now from the motor cortex, it sends information, this execution part. In the execution part, the motor cortex sends the information to the spinal cord through the corticospinal tract. Okay, and now that produces the action through the muscles and joints. And these action from the spinal cord either comes from the axial muscles, that is posture regulating, and the distal muscles, that is real action, the precise movements. Now, coming back, this is one component. The second component, the motor component, also provides the corticopontine or corticoreticular fibers, and through the reticular system, they will reach the spinal cord. So then also this motor cortex provide information to the olivary nucleus, because whatever it is, either copy here, maybe it may give a copy here, or a direct input to the olivary nucleus. Maybe we may not have so many fibers arriving, and the same fiber giving a collateral makes sense. So that olivary nucleus. For our understanding, I have drawn a separate arrow here. Now, this olivary nucleus, and then spinal cord also gives the olivary nuclear inputs, so that the olivary inputs are coming from various parts. Now, this one, the motor cortex also provide information to the spinal and the vestibular part of the cerebellum, the spinal, the intermediate and the flocculonodular lobe of the cerebellum. So that is this thing. Now, from here, the overall actions would be monitored. You can just see that this whatever input is given, that would be given back. Whether that is correct or corrective measures are taken place. This is one component. And then from the muscles, the proprioceptive inputs, they will reach the spinal cord. From the spinal cord, it will reach the cerebellum, either as a ventral spinocerebellar tract, as I was describing, or the dorsal spinocerebellar tract, they will reach the cerebellum. That is one component. The olivocerebellar, from olivary, inferior olivary nucleus to the cerebellum, that would try to make the correction, and that is intimated to the motor cortex, and that is intimated to the olivary nucleus, and that is intimated to the brainstem. So overall, the circuitry of the cerebellum is more complex than the circuitry of the basal ganglia, because it involves so many things, and because of these things, we are able to perform wonderful functions. Our actions are smooth and very, very much enjoyable. Now, coming back, just I read the cerebellar processing, whatever I'm talking about. Output of the cerebellar, deep cerebellar nucleus is excitatory to respective area. What are those respective areas? Vestigial nucleus, or vestibular nucleus, or reticular nucleus. In case of the vestigial nucleus. In case of the interpositus nucleus, it is to the red nucleus, collicular nucleus, and reticular nucleus. In case of the dentate nucleus, it is to thalamus and cerebral cortex. The Mossy fibers, they originate from various areas, excite the nuclear cell, and then granular cell. The granular cell in turn excite Purkinje cell to inhibit the nuclear cell activity, thus acts as a slow stoppage or slow break, dampening effect. This is dampening effect. Thus, the motion begins. There is an excitation of the deep cerebellar nucleus, followed by inhibition of the inhibition, thus dampens and prevents the overshoot during action. Any action in our body is pendular or is oscillatory. It's a simple physical phenomena. And this oscillatory movement, oscillatory action, just like any string and a ball moving here and there and here and there, if there is no resistance, they can go for infinite time. So in that case, so this has to be broken, and that dampening effect does that. The climbing fibers from these are corrective fibers from the olivocerebellar tract excite the nuclear cell, and then Purkinje cell directly to inhibit the nuclear cell activity. Abrupt break, a very strong brake so that stops the overshoot or the danger. This is about the cerebellar processing. The functions of the cerebellum include coordination of movements, that is the sequencing and the performance of the each muscle activity are coordinated, very well coordinated. Sequencing and scheduling of movements, that is the timing of movements. The feedback correction of the movement, that is a servo control mechanism. So that means it should not overshoot or it should not undershoot. So that the feedback correction, smoothing the turning repetitive movements, smoothing the repetitive movements. If I am doing like this, if this is very smooth, and sometimes what happens, this is not smooth. So that smoothing of the movement and linking the muscle tone, that is a alpha gamma linkage, that is the anterior part of the cerebellum, and the posture and equilibrium. So regulate the posture and the equilibrium component, especially the flocculonodular lobe. Eyeball movement, this is a vestibulo-ocular reflex, and this produces the nystagmus. Then motor learning and cognitive functions, because whatever I was trying to mention about, whenever we start doing actions, those actions are refined day after day. Day after day, as you keep doing the actions, it will become fine, smooth, and this fine smoothness of the action is because of those balance between the Mossy and the parallel fiber inputs. Then this is the anticipatory actions. So that means performing a precise ballistic actions. Precise ballistic actions, or hitting the keyboards of the computer, or definitely I consider the mobile is one of those important structure wherein we use a very small area, we perform it wonderfully. And maybe I should say that the younger generation is doing much better than the older generation because of the usage and particular skill you have developed over the period of time. You don't know how hard it is, but you are doing it wonderfully. Now, the assignments I'm giving the describe the connections and the circuitry of the cerebellum. I have given large part here. Briefly describe the basic working plan of the cerebellum. I have mentioned it. Then write short notes on cerebellum as a comparator and the dampening effects of cerebellum, how it dampens the actions, simple spike, complex spike, correction effects of the cerebellum. The reference books are the guidance, physiology, because where large things are, a lot of things are discussed in cerebellum. Then Candle's Neuroscience, the Genong's Review of Physiology, and Samson Wright's Physiology, because overall concepts I have trying to make and trying to provide may not be limited to these books. Maybe in addition, there are many other books. I have referenced them at a particular moment. Next class, we will continue with the signs and symptoms of cerebellar disorders. Okay, thank you.